very much. Happy to stay. Enjoy your beer. So I thought I will give you a biographic sketch. We're in 1962, late spring. I am a mere, not even an itch in my father's scrotum. I'm a spermatogonial stem cell. Somewhere round and biglary, which means I have 2 copies of the DNA that I inherited from my mom and from my dad. And on the way to becoming a sperm cell, I'm actually being put aside shortly after conception. I was conceived in 62 on a boat in the middle of the Atlantic Ocean, the Cristoforo Colombo, sailing from New York City to Genoa, as my parents were leaving the US after a 5-year postdoc that my dad did here. And once I was conceived, very early during fetal development, some cells were set aside to eventually become those stem cells, the germline. And the idea is that some cells are really they never die. The cells that made the sperm and eggs that made you, and the sperm and eggs that you make that might become the next generation, go on until the species goes extinct. So this notion of a sperm line, a germ line, and a somatic line, which is all the other cells of your body, their only goal in life is to make the next generation. So you can really argue that you are a sperm's excuse to make more sperm and eggs, depending on your sex. So I started out in the testes of my dad as a sperm, and it took me 2 months to mature. What did that mean? In the testes, the men, once they reach puberty, their germ cells awaken after 15 years of boredom, and are told, now it's time for action. You have to become a sperm cell. Sperm cells are totally weird. They actually lose half of their genome. So half of the DNA is tossed out. And now I'm a haploid cell. And I'm actually going to lose a lot of cytoplasm as well. I'm going to lose some weight. I still have my other half of the genome. I need that. But that's recombined. That unique combination of DNA has only existed once in the history of life. It is the reshuffled genomes of my parents and the double reshuffled genomes of my grandparents cut in 2. So it's a haploid cell. I will keep my genome, go back in my pants. And so I've changed, I'm haploid. And as I come about, I'm holding 2 sister sperm on this side and 1 sister sperm here. We are tetrad. 4 haploid cells, and we change. We become skinny. We lose the cytoplasm. We're still in the testes. And eventually, I grow a tail. And the tail is ridiculously long. I'm too scared. I'm about 200,000 eggs. So that's my tail. Yes. And so now I have a tail. I have a tail, but you might think, now the sperm is going to move. No, I'm not moving. I'm not moving. I can't move. I'm frozen, but I get pushed out of the testes. Pressure in the testes because you guys here are making 3,000 sperm a second. I'm being pushed out because there's more sperm being made. And I made it out to the epididymis. And the epididymis is the graduating academy. It's a long tube. It's as long as a parking lot. So in my case, it would reach from here to the Mexican border. So over the next 10 days, I'm going to be pushed from here to Tijuana. I arrive in Tijuana, I can dance, I can move. Not before. And all kinds of things happen. I forgot some important things on how to step here. I'm not ready to leave the testes quite yet, so let's go back. I'm still being pushed out slowly. And as I mature, very importantly, I have to be protected. So, now I'm a sperm. Okay, I get into the epididymis, and what is very important is already you see the white. The white represents my membrane, and it's totally packed with full of sugar molecules. Every living cell in your body is defined in part by the collection of sugar molecules that are on the surface. These sugar molecules are not encoded in your DNA. Only the enzymes that put them together are encoded in your But the precise flavor of sugar molecules I have on me now already tell the body this is a sperm. Very strange cell, the only cell that evolved to go from one body to another body. There's a few exceptions, you know, the Tasmanian devils, they bite each other and they can infect each other with this tumor. Well, that's another cell that figured out how to go from one body to the other. And you might have dogs, some of you, they like to lick their behinds. There's a wolf cell that is still alive, it's being licked from one butt of the dog. That's an exception. Sperm, on the other hand, are very respectable, usually not pathogenic. They have evolved to move from one body to the next. Okay, so I'm ready to graduate, but not quite. As I move through the epididymis, by this point, my genome has stopped expressing genes. I don't express any genes anymore. Any change that is going to happen to me, and many changes are going to happen to me, are due to these 10 days spent in the epididymis. I actually get dressed properly, you know, lab safety, so forth. I get another coat, even more sugar. Now these sugars are not made by me because my genes are silent. They're made by the epididymal cells of my dad on the Cristoforo Colombo, remember? I was on a boat. It's kind of boring, you know. So I get this coat, and once I have this coat, I become quite well protected. I need to be well protected because my journey will be into the female reproductive tract of another individual. I don't have the same MHG genes, I'm different genetically. You know, I could be perceived as an imposter, a parasite, a little bastard, whatever, you know. I'm protected. So as I go through, I'm still not moving, I'm being pushed by pressure. I get things like camouflage, you know, to try to hide myself. So that's one thing I get. That's enough. That gives you the idea. So I try to look invisible, but should I be perceived, I come in peace. I want to look really peaceful. You know, I don't want to threaten, because if you threaten, that's bad news. You get killed. Okay, that's the peace sign. But I have some other tricks. And many of these tricks have to do with glycoproteins and glycolipids that come from the cells of my father, are secreted by the epididymal cells of this long tube and put on me. They insert themselves on my membrane and they become me. And they have signal value. They will protect me from the immune system of the female, which is rather dramatic and not necessarily very friendly. So I have things that say, don't eat me. Things like CD47 that will tell the macrophages, hey, don't eat me. Maybe eat the neighbor, but not me. And then I have other things that say, I am you. I look like you. You can chill out. I'm not invading. I'm not a threat. And many of these molecules actually are glycoconjugates. So some really strange ones. They have a lipid tail. They're called GPI anchors. They have a little protein, and on the protein they have all these sugars. Even though that protein exists on white blood cells in your body, the type of sugars on that protein have only been described on sperm cells. So sperm cells are covered in chauvinist sugars that are teasing the female into not killing me. And as I approach the end of the epididymis, I'm still not moving, right? I'm just being pushed. I approach the end of the epididymis, I get a very special coating of extra protection called beta-defensins. Beta-defensins are proteins that usually punch holes into bacteria. You make them on your skin to kill bacteria. In the epididymis, there are special beta-defensins that carry a whole bunch of sugar molecules that look like the average cell in the female. And my entire body is coated with those. And once I'm fully coated, I start to gain motility. Because some of these defenses also make channels that allow calcium in. Now I move, nice and symmetric. I start moving, I'm ready to mosey, you know? I'm waiting for the alarm and so forth. And so depending if the situation is right, you know, and then you better hold yourself because you're ready to go really, really fast. The muscles of the vas deferens are your strongest muscles, guys. Way stronger than your guns or anything else. They are super strong and they will send you out very, very rapidly. But guess what? As you leave, that is not made by by your testes or your epididymis. It's made by your prostate, which has a good function before it starts growing. But you get this, the love potion, yeah, exactly. So you get coated with even more glycoproteins that will manipulate the female immune system. And finally, you arrive, depending on who the guy is and how long it's been since they saw each other. On average, 200 million of us arrive in the vagina. Normandy, woof. We're moving symmetrically. And it's weird because it's totally different. The pH is wrong. Luckily, some of the seminal fluid that came along buffers you. But really what I need to do is where's the cervix, where's the, there, let's go to the cervix. It's through the cervix. I forgot one bit, let's backtrack. Unfortunately, I have these sugars on me, which are good to be hidden, but things like the HIV virus have learned to bind these sugars. So watch out. Some of the sperms might be like a dragon ridden by, because they're about half your immune cells are neutrophils, the unsung hero of the immune system, those olfactory receptors I hope. It's like elevator music or new age music, and red carpets, and it's beautiful, and I can sit down, I can relax. In fact, female cells, ciliated female cells are holding me and allowing me to relax, and I can recuperate. And there's only, maybe 90 other sperm out of 200 million. We're a tiny little group that made it. I can chill out for a while, up to a whole day, and then things get really interesting. Design that also came, I never get there. And now I move in an asymmetrical way. So I'm capacitated. Being released, one of us at a time. I go race towards the egg, and this thing is going to etch. By the way, I hit a wall of gel. Because I have this, I can digest the gel. But the gel is full of yet other proteins and glycoproteins and hormones, and they make me crazy. They make me so crazy that I lose, that will hold the sugars in a species-specific way. That is kind of the secret handshake. I'm a human sperm, you're a human egg. Mammals have been known to mount things that are not exactly their species. So, this big burn, I get through the zona pellucida, and now the proteins on my head interact with proteins on the egg, proteins on my head, with proteins on the egg. This protein is called Izumo, named after the merit shrine in Japan, and the recently discovered protein in the egg called Juno, named after the goddess of love of Rome. They have a firm handshake. She pulls me in, and I lose my tail. Tail stays outside. And I make it into the egg. And that's why I'm here. About. And you could think that's the end of the story, but really it's the beginning, because this zygote has to become an embryo. The embryo has to land and make a placenta and be carried out for months. And many of the same tricks I told you about that the sperm play with sugar in non-human primates. It's known that if you ejaculate a couple times in series, the sperm number goes down quite drastically in our species. Some other species are used to the fact that every time you mate with a female, there might be up to 10 other males mating with the same female. Chimpanzees, for example. People have actually tested chimpanzees, and up to 6 or 7 ejaculation in a row, there is no change in the sperm count. They ejaculate 3 billion sperm each time. That's why their testicles are almost as big as their brain. So usually, if you can just, show me the testicle of a species, I will tell you their mating system. Relatively speaking, human testicles are much more on the gorilla range of things, which indicate that we might not always have been monogamous, but we have not had in recent evolutionary history very strong so-called sperm competition in the style of bonobos or chimpanzees. Well, it's especially when you carry certain mutations that interfere with spermatogenesis. What I forgot to tell you is that this was a research done with people who were vasectomized versus non-vasectomized males, it is the presence of sperm that really caused the outflow of leukocytes in the uterus in the female. Seminal fluid by itself does not convince the female that there's something to be done and take out all these unnecessary sperm. Whether the female chooses, what her criteria are, we all don't know. These are all interesting questions that dogs, offer themselves. Yes? Other questions?
Any other questions? I can bring you the mic.
Come on.
So then if you're a couple who's having trouble getting pregnant, say, should you really time when you're having intercourse because these sperm counts are decreasing so much?
Well, so that's two questions really. One is you're reminding me to comment on the fact that mostly in the industrialized world, there is a real trend of decrease in sperm numbers and sperm quality. That's almost certainly due to a lot of different factors, including pollutants, hormone disruptors, but no one has fingered the living relatives. People I know who have worked with chimpanzee fertility, you put a male and a female together when she is ovulating and they advertise it with a big swelling in the back, she gets pregnant, almost always. But in humans, it seems to take a lot of up to a long time to get pregnant. Even those 5 out of 6 couples that are fertile, right now it's about 1 in 6 couples that have problems conceiving. That also has to do with waiting longer until you try. So there's a lot of confounding variables, but there seems to be something off with human fertility, especially in rich nations that have all kinds of packages of the molecules we have newly introduced, how we live, and so forth. It's a huge question. It's of quite some concern because we have this paradox where we have 7 billion and going on the planet. So really we shouldn't populate more, but at a local scale you want balanced populations. You don't want just old people and no babies. So really there is a need to understand how we could alleviate the infertility issue.
I have a question, and then you said 1 in 6 couples. So if you split that couple up, both of them have the potential to be fine?
Not necessarily. So it's like the Anna Karenina principle. There's endless ways a family can be unhappy. And there seems to be endless ways that a particular couple could be infertile. It could be as simple as a compatibility problem. That's very poorly understood. One idea behind this type of possible selection that the female body is doing of sperm might be a selection for most compatible sperm, not fit or good-looking or sexy sperm. But given that this fetus has to live 9 months within your body and be accepted enough but not rejected enough, there's a very interesting idea that female mammals actually look for maximum compatibility in the gamete of the male. And so there it might be as easy as, having sex with another guy, that's sometimes socially complicated. But, it's known to happen. Not uncommon for a couple presenting at a fertility clinic that upon change of partner, there's suddenly no issues. So that is towards a kind of... Yeah, exactly. Yes?
Is it because of that I know it's like second cousins, or is there something where someone that's slightly more related than someone that's distantly related to me genetically is actually more compatible? Could it be that because it seems less foreign to the female that you're more willing to let it in?
So that's very interesting. That question has to do with outbreeding, optimal outbreeding or optimal inbreeding.
Yeah.
One of the costs of sexual reproduction is that you can hit the jackpot of a genome, right? That baby inherited a unique egg and a unique sperm with unique combination of DNA that have never existed before. It's super fit. It's like the best possible genius human you can get. What a pity to rip it apart again when it makes eggs or sperm, which is what happens with sexual reproduction. If you could, you would switch to asexual reproduction and let it clone itself. Then you have a whole lot of really, really good individuals. We don't have that luxury. Mammals are ovulating sexual reproducers. So there's a cost in sexual reproduction that rip apart co-adapted genes. One of the advantages of inbreeding is that you can keep them together. But one of the disadvantages, with a second cousin or something like that, there is people who study genomics across the planet, have shown a very nice effect of cultural beliefs about what is inbreeding and what is not, in that there is an elevated level of long stretches of DNA without recombination from the Middle East to Malaysia. And the map fits perfectly with the prevalence of first cousin mating or uncle-niece mating. So that would be an example of a human culture and a human belief system that is shaping the genome by who can have kids with it. What about monkeys? Which monkeys? There's about 300 monkeys. Choose your favorite. No, it looks like in animals, well, across all primates and across most species known, there seem to be mechanisms of inbreeding avoidance, usually behavioral. So behavioral that the famous case of kibbutzim kids or little sisters in Taiwan, where parents can kind of adopt a girl who's supposed to become the wife of the little boy. Growing up together, spending childhood together is a really bad idea if you want to have a healthy sexual life later as a couple. Because these inbreeding avoidance mechanisms probably kick in, that you recognize each other as kin, and there is something off with you now being mates. So I think that's very often mediated through who migrates and avoidance. Like it's not unheard of that young male chimpanzees try to mate with a mother. They usually leave them up and try everything they can do to stop them from that. So biology is never 100%, and human history.
But recent research has said that paternal age is also important for the fertility rates and the chromosomal abnormality. But for the mothers, the eggs are in the ovary for long years, but the fathers, the sperms are made very often. There's I don't understand why and where they got those chicks and why they.
What causes the high mutation rate? So it's absolutely true that paternal age introduces more mutation, but it turns out even young men introduce more mutation than young women. The first type of mutation that scientists could study was chromosomal anomalies, because you can stain chromosome and see them under the microscope. You can't see DNA under the microscope. So people were able to find out that moms over 35 years old tend to have many more kids with chromosomal errors. Only 2 of these errors are viable, everybody else is lost. It's now clear from whole genome sequencing of father, mom, and kid that the father contributes almost 4 out of the 5, about 50 total new mutations that happen in the parent. Out of those, 45 of those 50 happen in dad's destiny. And with every age the father ages, he contributes with every year, on average, he adds 2 new de novo mutations. So you have clowns like me who have kids in their 40s. My 2 girls got 2 times the average rate of de novo mutations because I had them when I was in my 40s, which is another example of the cultural behavior that is changing the germline mutation rate in humans. So culture can be very powerful, even though it's not genetic. You're absolutely right. So paternal age is a risk factor for autism, is a risk factor for all kinds of de novo mutations that happen in the male, which smacks of this notion that males propose and the females dispose, which might be part of the answer why you need 200 million sperm to fertilize 1 egg. I mean, it's an embarrassing question that I cannot answer in my tenure. Like, I'm a biologist, I studied biology all my life. I have no idea why it takes 200 million in us, or 3 billion in the chimpanzee sperm to fertilize 1 egg. Why is that? I think it is linked to the fact that most of the sperm are some kind of tryout free. That properly get eaten up by the leukocytes, and should they make it to the egg and something is off, the female still has the option to not let the blastula implant in her endometrium, and then it's just slightly delayed menses, no investment.
That happens in the stem cells.
Pardon?
That happens in the stem cells.
What happens in the stem cells?
That mutation.
Yes, the mutations happen during spermatogenesis, and the chromosomal mutations, it is believed that you can easily lose or gain a chromosome because making eggs is an asymmetrical division. You end up with only 1 big egg and 2 colobuds. And so this asymmetry makes the female gametes prone to make mistakes with the number of chromosomes. Whereas division to make sperm is totally symmetrical, and that mutation is rare in males. All the other mutations, huge chunks of, 1 million layers that fall out. Happens all the time. And most of the time it doesn't cause a thing. You have a couple of million base pairs missing, and I have a couple of extra copies, and he has a couple of extra copies. Most of us are lucky, it doesn't affect us. But sometimes you lose a couple of really important genes, and then weird things happen. Yes?
How much of infertility do you think is heritable? So like, these people going through IVF, how many children do you think are going to?
That's an IVF question. How much infertility is heritable? In some cases, one of the very important reasons why some men make sperm that cannot move is called azospermia, so normal sperm count, but they never move. You can throw anything you want at them, and they don't move. And that's a mutation on the Y chromosome. So if these men present a fertility rate, 50%, now that their sperm don't have to move anymore. You can take sperm long before they put on their helmet and their glasses and their don't eat me button. You can take a testicular biopsy, take a sperm, and directly inject it into an egg, and you get a healthy baby. Which is kind of a paradox. You're bypassing a whole bunch of stuff. Maybe the baby seems healthy, and we don't know yet. Wait a couple of decades and we'll know. But the men that have azos, so you're actually directly passing it on. And you might be doing that with in vitro babies. And the vast majority of them, the oldest one is only 37, they seem totally okay, which is great. But the experiment is ongoing. Rapidly, people are pushing the frontier of proof, making window.
Right.
Yeah.
Why is that?
Actually, in the fallopian tube. And there's some very grim evidence from World War II with regard to when in her cycle is the female receptive. That shows that, with the exception of 4 or 5 days, women, and this is from reports of German women raped by GIs or Russian soldiers, pretty much any day of the cycle, women got pregnant, even through rape, except for about 5 days out of the whole month. And whether that reflects that the sperm gets to the egg, almost by definition, there must be some reservoir we don't know about. Some niches, maybe somewhere in the cervical crypt, in a buffered piece of mucin, you have a couple of sperm that survive. It wouldn't be unusual. A lot of insects mate once, like a queen bee will mate once on a nuptial flight, and then she uses those sperm for several years and just deploys them as needed. So, but in our species, embarrassingly, nobody knows. But it's almost certainly not as clear cut as your next textbook is going to be. Which ones? Which species? So sadly, I have to report that if anybody has an idea how we can get a mouse to ejaculate, I will pay you quite a lot of money, because then I don't have to kill mice as much as I have to. Right now, to study mouse sperm, we have to sacrifice the male. So we have to submit an ICAC report. We have to justify why this is important research, and then we get the okay to sacrifice a certain number of male mice in a humane fashion, putting them asleep and subject to starvation. In humans, it's very easy. We just have to find volunteers. The problem becomes that you have to be, I have to tell you that, you have to file an IRB. Were I to study my own sperm, Leuwenhoek, the guy who developed the microscope, they say he was in Holland, he knew how to make lenses, he made this great little microscope. They say the first thing he looked at was a drop of pond water, and he saw these animalcules. We're not quite sure whether it was a drop of pond water or a drop of ejaculate. He found sperm very, very quickly. I mean, boy, it's a huge thing, and people say, oh, that's where life is, and other people say, no, it's the egg that makes life. You have spermists against ovists, that say, no, sperm is some people classify sperm as a parasite, as some kind of nematode, then what do you both do? So, were I to try to do what Leuwenhoek did, that would be illegal at UCSD. I have to have a institutional review board approved protocol that explains how I study human sperm, including my own. And then I had to put down what the risk was, because they want to risk management. Like, okay, what is the risk of having somebody bring me a sample? I was very tempted to put something about like what carpal tunnel syndrome. So I said it's embarrassment. And that worked, so they were happy with that. So I had to convince them, the volunteers I contact are people that cannot be my graduate students because there is conflict of interest. They have to bring samples somewhere where nobody sees them, it's all incognito, and so forth. And then the chimp sounds, which is my favorite, is I thought I had it down. I made a wonderful, model out of latex of a pink chimp bottom. I brought that to a chimp facility in Arizona, and I held it in front of these chimps. And one chimp, I took some straw and put it in the stick. They were trying to look on the other side if there was a chimp on the other side. And then I talked to a couple of old people who actually had done it all already in Atlanta. I said, "Oh, just take a piece of PVC pipe about this long, put a little bit of latex tube in it, water in between, and just hold it there." Training with a little bit of some candy corn, fried, threatening. And then all I had to do, actually I couldn't collect it. It was a female caretaker that usually had the males come up and present erect whenever she showed up. So she just held the tube there. Before the no liquids, right? So I'm going through security. And I was like, holding them to ask them, what is this? I said, oh, you don't want to know. And the last thing so many times I've had chimpanzee sperm in tubes next to each other. And you spin them down, so they can separate seminal fluid. Sometimes you wash them, you rinse them out. It turns out chimpanzees make a plug. They put in 3 million sperm, and the latter part of the seminal there completely blocks the cervix from the next male. Except if the next male is a little bit better endowed, and a little bit longer, it will displace the plug and then fit this plug there. Well, another evidence that humans don't have a lot of sperm competition is seminal fluid in humans doesn't make a plug. Same thing with bonobo, chimpanzees, macaques, all the species of primates where there is a lot of competition for the same egg between their sperm, but they then plug the whole thing. So in a tube, the chimps can swim so much that after you spin them down, the male, they have a nice pellet that is all clear. You take the chimp sperm out of the same species. Good.